So my name is Mary Mader. I am the co-chair of the MMV ESAC. My specialty is into the discovery sciences, and I've been affiliated with MMV for, amazingly enough, since about 2014, so about 12 years. Where did the time go? I myself am a medicinal chemist. And so here today we're going to be talking about transforming drug discovery with new technologies. And our goal for this session is to explore how MMV and its partners are working to transform how we identify and develop or optimize. When I say develop, I mean optimizing the small molecules that end up being delivered into the clinic. So what I want to do is encompass current tools that we use for drug discovery, including ones that are developed by MMV and its partners, how these tools have evolved over time, especially from the perspective of drug discovery and understanding drug resistance, and then the role of collaboration in the development of these tools, and you'll see that illustrated here today. So our panelists and our presenters are actively involved in malaria discovery, and their perspective on this then is critical in describing the state of the field and what their future plans are. So we're going to start with two presentations, first from Jeremy Burrows and then Dave Hallet, and we'll have a short video from a colleague who couldn't be here in person today, and then we'll begin our panel discussion. So Jeremy, if you'd like to come up. This is Jeremy Burrows, who's the head of discovery at MMV. He'll be heading to the Liverpool School of Tropical Medicines next summer as a professor of global health therapeutics, and he'll be talking about accelerating the public health medicines of the future, democratizing machine learning for public health.
Thank you very much, Mary, and good afternoon to you all. So through today, we've been hearing about the need for next generation antimalarials, and from a discovery team perspective, we've been thinking about how new technology can be brought to bear, in particular, artificial intelligence. So I want to start with a question that I want you to just think about. What's your perspective on AI? So perhaps you're someone who is super excited by anything new and wants to leap headfirst and try it out. That's actually my sort of default position. Or you may be unsure. You may just feel that you just don't know anything. You're going to need a lot more information before you're able to give an opinion. Or you're a downright skeptic. You're not going to believe any of this rubbish until you see hard evidence. Okay, so I'm going to actually give you our opinion at the end of the talk. But I have to say that to start with, we began in the middle. We were unsure. We really knew very little. We needed to learn. And it's been a journey of discovery. And I'm very grateful that two of the individuals, Dave and Caroline, are here today. They've been hugely generous in sharing their learnings, helping give insights around really what is realistic and what is not. And obviously, it's a dynamic situation, so things are changing fast. I've been really inspired by their commitment to new technologies and ways of working. And obviously, they're here. They're willing to engage in public health. So, thank you to them both. And their input has helped us to articulate a vision of a world in which state-of-the-art AI equips all working in global health to deliver candidate drugs addressing the areas of hardest unmet medical need. Okay, so how are we going to do that? What's our strategy? So first and foremost, we're going to learn. We're a small PDP. All that we do is in collaboration. So we're going to learn and collaborate with the best, not least because malaria deserves nothing less. We also want to make every molecule count. We can't synthesize every compound that we want to make, but what we can do is we can make choices about what to make so as to maximize quality, maximize learning, and maximize the use of resources. Equally, we can't test every compound we make in every assay. But what we can do is we can make the most of our data by creating predictive models that we can then apply to virtual structures to actually aid with choice about what to make. And there's a huge inequity in access of these technologies in different geographies of the world. So we are passionate about co-creating and equipping scientists working in global health to democratize these technologies together. And as with every strategy, and I learned this from Tim many years ago, it's important to focus. We can't do everything, but at the same time, we want to be ambitious because of what's at stake. Okay, so before I let you know what we've actually done from an MMV perspective, let's just think about some of the things that AI is good at. So first of all, AI can be trained on text, and I suspect that there are people in this room who have used large language models like ChatGPT. So we know that predictive text, reformatting, collating, summarizing is really good. AI can be trained on patterns, and anyone who has facial recognition on their phone or their laptop, will be very, very familiar with this. And AI can use and it can process numbers. So if we are going to make the most of AI in drug discovery, first of all, we need to ensure that we can encode all of the science that we're doing in these sorts of descriptors. And actually, that's pretty straightforward. If we think of biology in the context of DNA, RNA, amino acids within a protein, these are all coded as single letter text. And the text and numbers and brackets in black, this is something called a SMILES string. It's a way of encoding a chemical structure in text. So this is all good. Obviously, huge variety of patterns that I could have chosen: the pattern of a protein structure, the pattern of organelles within a cell, or the patterns of electron distribution around the nuclei of atoms within a molecule. All of these can be source of areas for training in AI. And of course, in terms of numbers, we can convert biology and chemistry into numbers. For example, an IC50 from a biochemical assay, or encoding a chemical structure in ones and zeros in a so-called, a chemical fingerprint. Okay, so to start with, AI is good with big data, and we've done a lot of phenotypic screening. We've screened over 10 million compounds with partners, and what we've been able to do is work with computational chemists independently at each of our pharma partners and others, who have been able to model their data to basically define whether it's active or inactive on their 2 million compound libraries. And then we've been able to bring these models together in a consensus, confidential, safe way, and put them on the Chembl website, where anyone can go there for free and can upload a SMILES string, a virtual chemical structure, and get a number. And that number is basically a prediction of the likelihood of that compound will be active in a growth inhibition assay of falciparum malaria. And what James, Dom, and Elodie and the team have done is they've done a really nice experiment where they've acquired compounds through sort of random picking, and they've acquired compounds based on use of the model. And they've shown that the model increases the hit rate by tenfold. And this is significant when it comes to really thinking about what compounds we could buy and we could test. So this is all, I didn't actually say it, but this is all a model called the Malaria Inhibition Prediction Tool, which is MAPE for short. So we've talked about pattern recognition already, and we can now move to cellular pattern recognition. And we've got a lovely project with a Japanese company called LPixel, the University of Dundee. In fact, Marcus is in the room, and so you can grab him afterwards and find all the details. And this is funded by GHIT with involvement from MAV, from Marina, from Benoit, from Brice. And what this project is doing is we are creating models to recognize the parasite pattern that is linked to a distinct compound's mode of action. And this is actually really exciting because we still do a lot of phenotypic screening. And what this basically means is that at the end of this process where we've built these models, when we have confirmed actives, we will be able to look at the particular pattern and get an idea as to whether this is a new mechanism or a new pathway or not, which could influence actually the prioritization in early stages, potentially from reducing it from months to days. We're also passionate about open access pharmacometric tools, and so this is really the idea of equipping teams to focus on holistic optimization of parameters within lead optimization. And in essence, what these tools are doing is enabling teams early on to get a prediction of human pharmacokinetics and an early dose prediction. And our sort of flagship tool is called MMV Solar. And what you can see in this graphic, for a particular simulated dose, you can see the tool has received a lot of data from the project and then predicted the human pharmacokinetic parameters. And so in black, you have the compound concentration time course. And then due to the concentration response on the parasite, either in vitro or in vivo, you see the parasite time course over time. And this again has been transformational in the thinking of projects, because it enables a team in early LO to understand how far away they are from delivering a candidate drug and actually what properties they need to optimize to actually get there. But the main area of focus for us at the moment is in medicinal chemistry drug design. And so we're working with a company based in London called Deep Mirror, and AstraZeneca are giving kind support, and with funding from the Gates Foundation to deliver a tool called Drug Design for Global Health, or DD4GH for short. And this is a project that is being led by Andrea and Max from Deep Mirror, Benoit, Ola, Dom, and Jeremy. And this is a tool that is going to be delivered in March 2026, so we're not there yet. It's going to be free access to all those working in global health, and we believe that this is going to democratize AI in drug discovery design. In fact, we were in this very hotel, a few floors down, 2 weeks ago with almost 50 people, with a workshop exploring machine learning, AI, and drug discovery, and particularly the focus on this tool. Really co-creating for the future needs by the time of delivery. And we have another workshop in Ghana with Richard in the beginning of December. What this tool will do is I believe it will improve the quality of compounds with the potential to have impacts on timelines and cost. Now, what does this tool actually do, though? I guess, first of all, this is a tool designed for medicinal chemists. People like me, I'm not a computational chemist, I'm not a coder, an engineer. I need things to be really, really simple, intuitive, and straightforward. So what this allows me to do is upload my data, compound structures and data. The data is modeled. That's number one. Then we have generative design, so the tool creates virtual chemical structures around the front runners, which are then scored against the model. And then in 3, we can actually triage effectively based on the target product profile, on the goals of the project. To 4, where ultimately we need to make choices about what sorts of compounds to actually make. And those compounds are selected not only that they will improve the project, but critically that they will improve the models. And so by getting those compounds made and tested and the data fed back in, we close the learning loop, we improve the models for the next generation of design. But it doesn't just all stop there. We have a grand vision for the future. And first of all, we've written a concept note and shared with the foundation, because they invited us to do this, for a second version of DD4GH, which is going to incorporate what we call 3D-driven design. And so this is really where the technology, the co-folding technology comes in. So this is sequence to protein structure, to binding to ligands and cofactors. And this has all been led, this has all exploded after the innovation of AlphaFold and the various iterations, and newer technologies like Boltz-2. And really the vision here is that within the tool, we will be able to actually look at the scoring of virtual structures against wild-type mutants and orthologs. So in effect, we can optimize for potency, resistance, and selectivity all in one go. I think that's really exciting. We've got lots of plans to actually further leverage our data, and we want to reduce the time it takes to actually get compounds actually made or acquired more synthesis-aware modules really within the tool. And then finally, we talked a lot about the alternative target product profiles from oral to long-acting injections. We want to encode 4 long-acting compounds in period, but specifically for long-acting injections into the tool. Okay, so I promised to tell you where we got to against those three options. And the first thing to say is actually it is really important to have a degree of skepticism and to be wary because there are many bold claims that need to be substantiated. And I've seen all sorts of things of what people have said, and it just hasn't gone anywhere. And we want to validate and learn the limits and potential of new models. So it really is critical to have a human in the loop and to use our brains. But of course, it's a constant, there is a constant need to learn and to implement outside expertise. But it's not just about actually doing that in the context of new tools, but also new ways of thinking and ways of working, because the world is changing and this is very different and it has an impact on those. And I do passionately believe that this is an extremely exciting area for the future that's going to enable us to make the most of all of our data, make every synthesized compound count, to democratize AI for LMIC scientists, and co-create tools that will transform how we work. Thank you for your attention.
First of all, big thank you to Jeremy for the kind invitation to present to you today. Let's see how receptive this audience turns out to be. Look, I don't have time in 15 minutes to kind of go through a whole list of things that and techniques and technologies that recursion and how we apply it to modern drug discovery. But I thought, given the audience that I have in front of me, let's choose a real-world application that we've been working on together with our friends and colleagues at MMV. So, I think the central tenet at Recursion, I think the central tenet actually at a lot of places is that what we're trying to do, the way we're trying to apply artificial intelligence and machine learning is to actually accelerate the rate of which we learn. I would put it to this audience that some of the greatest inventions and progress that humankind has ever made has been in an environment where kind of multidisciplinary people and therefore the data has come together and they've been able to iterate very quickly. Now, unless you believe that when NASA put Neil Armstrong on the moon, that was the biggest deep fake ever. But if you actually analyze how they got there over that 10-year period, it was the Mercury program, it was Gemini, it was Apollo. They didn't just decide one day, oh, I'm going to basically take off from Cape Canaveral and go to the moon. It took them a decade of iteration. And actually, if you look at any inventions over time, I think hopefully you would agree that that's typically how humans have progressed. Create an ability to rapidly iterate and prototype, and then learn and then move on. I think the other thing that kind of Jeremy touched upon is that I think, and I see why there's a skepticism as well, I think ultimately the belief behind the use of AI and ML is like, how do we reduce the cost of R&D? How do we accelerate R&D? How do we get R&D to kind of succeed more than it fails? Because that's obviously the source of a lot of underlying costs. And I believe that AI and ML can help here. These are bold claims, and clearly only time will really tell if that's going to be the case. So, talked about learning. For those members in the audience, I think there's a few here that, like me, are kind of one-time medicinal chemists. What I've described in this diagram is typically a learning loop for small molecule drug discovery. Whether you're using your human brain or whether you're using computational systems, it's always the same. At the outline of a project, you're going to assemble what data you know about a target. That might be a protein structure, it might be nothing, actually, it might be an AlphaFold structure, it might be a predictor structure. It might be ligand data if you're lucky, like the data, the sources that kind of Jeremy spoke about a few minutes ago. But it's all about kind of assembling a data set in order to try and build a predictive model. We do this all the time as humans. Human beings basically are generating models all the time in our heads. We just don't realize we're doing it. It's kind of almost subconscious. All we're trying to do here is reduce that to kind of some kind of systematic practice. So the way we're applying AI/ML in what we call a Centaur suite is that, so at the start of a project, the first thing you need to do is I need to design some molecules. But I need to be able to make them. So we use synthesis-aware kind of tools to try and predict not only the molecules that will deliver the properties we need, but can you even make them? A lot of property predictions at the start of a project, kind of physical properties. Will they actually bind to the target? Will they modulate the target? We'll typically maybe score tens, if not hundreds of thousands of kind of molecules computationally. But the whole point of accelerating this is to predict more, kind of to make less, to test fewer. Not sure if you're aware that typically for every small molecule that a company will make and then physically synthesize and then do a simpler round of testing, so we're talking like basic DMPK properties, pharmacology, you're talking about $10,000 per compound to get that data point. So if you think you're making 100 of those, you can do the math, you're making 1,000 of those. That's kind of why modern drug discovery can be so expensive. So if we can figure out a way in this kind of learning loop to both to make fewer compounds because our predictions are better and to test fewer compounds, there's a pretty good, and to learn from that in this kind of environment. And that's how this works. So we make compounds, we do actually test them, we ingest that data back into our systems, the computational models are updated, and they should improve as a function of time, and we move on from there. One final point before I go into the example. So it is important that, or we felt it was important that any predictive platform has to be both data and model agnostic. You don't know what data is going to be the most valuable at the start of a project. If you're lucky enough, it might be a crystal structure, but if it's phenotypic data, you don't even know what the target is to start with. So you need to be able to ingest different kinds of data, text data, alphanumeric data, could be high content images. You also don't know what the best model is going to be at any one time. So the statistics, is it a regression model? Is it kind of a deep learning? Is it neural networks? You don't know this. And also what we're seeing in today's day and age is that kind of models are just becoming commodities. And so everyone's kind of improving as a function of time. And so we built a kind of platform that integrates all of that. And ultimately what we found is that typically in the successful drug discovery projects, and we have a few now that are actually probably about over a dozen now that we've taken into human evaluation, typically it takes about 10 to 15 iterations in a given chemical series to go from initial starting idea to compound or to candidate. And typically we'll make about 300 to 400 molecules during the lifetime of a project. So let's go on to the application. So a big thank you to the Gates Foundation that actually co-funded this particular this iteration of the project. So first of all, what's a bispecific? So it's a small molecule, so it's not to be confused with a bispecific antibody. That's not the same. And what we're trying to do here is design in a single small molecule footprint a molecule that can deliberately and selectively modulate two different protein targets at the same time. So this is not the same as like a fixed dose combination. That's different. That's kind of like two different molecules. So this audience knows better than I do what that looks like. But they were independently developed. They were independently often taken through toxicology. They were co-formulated. What we're trying to do here is to try and design in a single molecule, and the benefits of that are obviously kind of cost of goods in terms of kind of future development. But they need to maintain their small molecule footprint. And at Recursion, we've actually had some success with this. So we currently have two molecules that were designed off our platform. They're being advanced in psychiatry by a partner. We've also got a few at the peri-IND stage, again with partners in both metabolic and inflammatory diseases. So the idea here, when we spoke to the Gates Foundation, is both in the malaria. We try this in the TB environment as well, with less success, I have to be honest. That is, can we build a scalable platform that can do two things? One, can we, first of all, create a large data set with the help of the folks from people like MMV to allow us to build a predictive platform that might then allow us to then, first of all, select, well, out of the malaria kind of genome, which targets do you think you can actually modulate at the same time as a small molecule? And ultimately, can you actually design molecules against that and show that they both inhibit their targets, that have phenotypic activity? And of course, one of the benefits potentially of a bispecific is that if you're modulating two very different pathways at the same time, particularly if they're essential, do you slow down the rate of resistance? So here's the framework. In order to do this piece of work, and this is kind of one of the gifts we're able to give back into the community, was like the actual data and the model infrastructure that sits underneath this. Step one was to, first of all, again with the help of people like Paul Willis and kind of Jeremy, to actually ingest the data that's there and curate it. One of the big issues with kind of machine learning, and as our colleagues in the audience will know, garbage in really is garbage out. One of the biggest things you need to do in terms of machine learning is actually have a data set that you can actually build models from. And so a lot of the work that was done here was actually both computational and manual curation. Picking out kind of like understanding which targets are actually essential, which ones actually have any information on at all. We then took that into, we then built models off the back of the known data, and this led to this kind of the ability to kind of pick dual targets. You can't just pick your two most favorite proteins from a proteome and design molecules against them. It doesn't work that way. It's like kind of, even though there's a lot of degeneracy in protein structures, you have to try and go out and predict which ones actually might be applicable here. And ultimately, then we generate molecules that have both drug-like properties or predicted to have drug-like properties and might modulate the two targets. So that's what we did here. So this is if you like, the big piece of work at the start, building the foundation. So we created an infrastructure to bring in both training data and then to build kind of models. So what we're doing here is first of all, against any single target where ligand data exists, can we predict that? Kind of like, so can we build our models and can we QC them? We then would then run generative design matrices against these protein pairs. And you generate these kind of interesting things called butterfly plots. So probably not the clearest here, but essentially what you're looking at is that, for those of the audience, on the X and Y axis in both red and blue dots, that's real experimental data against two individual targets. And we train our models on individuals. And then you then ask the model platform, can you design molecules that are predicted to kind of modulate both? And more importantly, can you actually tell the difference between decoys? So the decoy molecules are basically the green molecules in the bottom left, and the yellow ones are basically where the model predicts it can design into that space. This is a butterfly plot that we look at. They're not always like this. Sometimes the models are just not highly performant enough, and basically it's very difficult for the model to even differentiate from random. Once the genes were curated and selected, basically we overlaid the chemical data that was provided, like things like Kemble. Some of our founders actually kind of were the brains, if you like, and the power behind Kemble in the first place. And then we could do kind of pairwise generative steps. We haven't made anything yet. This is all prediction at this stage. And then what we do is we kind of filter based on what's the confidence in the model that we can actually predict, we can simulate, we can inhibit both two targets at the same time. The butterfly plots is one way to visualize that. A Shapley value is a value taken from game theories. Basically what it allows you is that it's a way for you to kind of quantify the individual contributions from two or more pieces of data. We'll predict drug likeness score, we'll predict how easy on it's compound to make, we'll overlay that with any 3D structure information we know about the protein and move from there. And in the end, we move forward with 4 target pairs that we took into both synthesis and testing. These are the 4 that we picked out in the end. Some easier than others, a priori. So like pair 3, 2 kinases. You go, Dave. Kind of kinases are pretty easy to drug. I can design 2 at the same time. Yeah, I would agree with that. But try and make them selective against the rest of the kinome. That's not straightforward. We've got dihydrofolate reductase there. We've got myristoyl transferase. These are all targets you knew about. Our friends at MMV and the Carewells helped us to pick these because we're looking to try and find genes that themselves were essential to the blood stage of the parasite, but also were not in the same pathway. Remember, we're trying to get the synergy between inhibiting and modulating 2 pathways. So in the last few minutes, what I'm going to focus on is what we did in kind of what we call phase 0 and phase 1. This is essentially kind of hit finding. Can we even find hits and then validate them in order to try and to prove whether this is even doable? And I'd like to thank the team at MMV because, somebody said earlier, it really does take a village to do this. Recursion and myself, I knew very little about malaria when I came into this project. I know a lot more than I did 4 years ago, if I'm honest. But it takes the funding from Gates. It takes the support from people like Jeremy And so we put an infrastructure in place to allow us to actually experimentally kind of test and try to prove this out. So this is what a typical design cycle would look like. I'll try to reduce it to kind of a schematic here. So you start from in this case, probably about 1 million data points against NMT and PKG. So it's quite a data-rich environment. Using very different generative methods, because there's more than one. So we're using either what we call reinforcement learning methods that we internally call Rogue, or a more evolutionary design. We generated computationally about 15,000 molecules. We then applied kind of drug-like property filters against that, and that reduced it down to about 11,000. You then apply a much tighter constraint. So now you basically take it into do these molecules, are they predicted to to bind into either a predicted or a known structure of the protein? That takes it down to about 1,500. We then reassessed for synthetic tractability, looking at building block availability. And in the end, we basically made about 50 compounds, but we assessed millions. Some data. So that was all predict. This is the reality. Sometimes it works and sometimes it doesn't. So what you see let's show the two on the right first. What you're seeing here is like a single round of synthesis. We could make some compounds that are in the kind of 10 micromolar range, but not many that basically made it kind of above, say, single digit micromolar from the first design cycle. But on the left, you see two pairs where we kind of got initial evidence that actually we're making progress here. So looking at pair one, what you can see here, again, you see a distribution you'd expect at the start of a project. But you can see we're kind of routinely kind of both predicting and then making compounds that were in the basically 1 micromolar to 100 nanomolar space. We would then characterize those. So again, this is all based on prediction, but this is real experimental data. We set up assays for both biophysical assays, so proving do our molecules actually physically bind and engage the target that we predict them to, and actually are they also functionally active, because they're enzymes as well. And the short answer to that question is yes, is that these compounds were active in both biophysical and enzymatic assays. In the first round of assays, I would say that these compounds are slightly more biased towards PKG affinity than to the transferase, but they were dually active. We also wanted to check about ortholog selectivity, because if you think about this, one day we might want to take these into human beings. So the more selective we can be, the better. And so what we're showing here is that in the case of both the kinase and the transferase, we're probably about at least 10-fold selective, if not better, against these. That's still an area obviously to improve, but remember, these are just hits. These are not drug candidates. We're also able to show that the phenotypic activity, so what you're looking at here is a 3D7 kind of parasite assay. And what we're able to show here, again, a kind of a range of phenotypic assays. So in some cases, kind of micromolar or better, but in these early stage compounds. And we're able to show the way we've color-coded these. So on the left is phenotypic activity on the x-axis, and then on the y-axis, and color-coded are and the deeper the orange, the more potent at the target. And what we're able to see here is that the phenotypically active compounds were actually active at both targets. And we have other additional data that support the notion that this wasn't just a PKG effect, it was actually the activity was driven by both. Remember, we're not just talking about ligands either. So again, this is real data, not predicted. So what you're showing on the bottom left is two routine stability assays. So human microsomes on the y-axis, human hepatocytes on the bottom. The more stable compounds are basically bottom left. Again, you can see a kind of a range of activities from kind of very stable in a human system to less so. But these are quite good from a model building perspective. If you look on the top axis, on the top right, what we're looking at here is a matrix of physical properties versus stability. And again, we've got compounds that have they're not brick dust. Somebody said that word earlier today. Some of them are, because it's very early in a project. But basically, we've got compounds that have got decent in vitro ADMET properties, and we also did kinase profiling. These compounds are actually got good selectivity against the rest of the kinome. And that's where we got to. We identified target pairs using kind of this methodology. So before we started designing molecules, we picked out and predicted what are the target proteins in the parasite kind of genome we might be able to inhibit. we developed and validated some novel hits against those. We actually, it's not shown today, but we've kind of, Jeremy's team helped. We showed that we've got some initial data in vitro that at least is indicative of this idea that if you modulate two essential targets at the same time, you do slow down the rate at which resistance kind of is formed in vitro. So that's a good thing. And more importantly, I'm delighted to say that literally today, I actually signed the DocuSign agreement so that Recursion have basically been awarded a $7 million follow-on grant to work together with Jeremy and the MMV team to take this work from the hit stage, but try and forward into advanced leads. So I think I agree with what Jeremy said. I'm a skeptic too. I come from 25 years of drug discovery, but I truly believe in this technology. Yes, it's overhyped. I get that, but it does work. I use these technologies every day in my life. Yeah, trust but verify. That's true in any walk of life. But I do fundamentally believe that kind of machine learning is here to stay. I will say that basically scientists that don't use AI will be replaced by scientists that do use AI. I think that's just a fact. I don't think AI will replace scientists, though. That's not the same. Yeah, just to be clear. And I think kind of Jeremy, he's a skeptic. He came to work with us for 6 months. I think hopefully he enjoyed his sabbatical with us. And the Gates were kind of sufficiently impressed with this initial data that they actually gave us a follow-on grant. And I will stop there.
Next, we have a short video from our colleague Zoleka Ngcete, who is a program manager for Strategic Health Innovation Partnerships at the South African Medical Research Council.
Developed in partnership with MMV and brought into the clinic. Hello everyone, I'm truly sorry I can't be with you in person today, but I'm grateful for the opportunity to share a few words with you virtually. The partnership between the SAMRC and MMV spans over 2 decades now. Together, we've achieved some incredible milestones. One of the most significant is MMV-048, developed in partnership with MMV and brought into the clinic through first-in-human trials conducted right here in Africa. This achievement not only broke new scientific ground, but also signaled Africa's growing leadership in global health R&D. This partnership has also been instrumental in building the continent's capacity and capability to undertake drug discovery. MMV's support for technology and skills transfer to H3D has enabled the growth of local expertise and infrastructure. A standout example is the recently established African Drug Discovery Accelerator, supported by the Gates Foundation and LifeArc, led by H3D and supported by MMV. And this program is helping to shape a new generation of African-led innovation. These advances have been made possible thanks to catalytic financial support from the South African Department of Science, Technology and Innovation. In their decadal plan, malaria has been identified as a key focus area for the next decade. This aligns with the South African Department of Health's new malaria strategy, which reiterates our country's commitment to eliminating malaria by 2028. These national frameworks provide a strong foundation for sustained innovation and impact. I'm especially excited about today's focus on machine learning and artificial intelligence. Congratulations to Kelly and the H3D team on their collaboration with Asilia, an inspiring example of how AI can accelerate discovery and deepen our impact. For South Africa, AI offers a transformative opportunity to leapfrog traditional barriers and drive locally relevant solutions. As we look ahead, I remain confident in South Africa's unwavering commitment to defeating malaria. We are grateful to MMV's for its partnership, as well as other key partners who have been part of our journey to build drug discovery capability in Africa, including the Swiss Tropical and Public Health Institute, the Technology Innovation Agency of South Africa, the Gates Foundation. Through science, partnership, and innovation, we will continue to push boundaries and deliver hope. Thank you all, and I wish you a productive and inspiring meeting.
I'd like to invite the panelists to come up to the stage so we can have a brief discussion over a couple questions around machine learning, data, artificial intelligence. We've met Dave and Jeremy. We also are welcoming to the stage John Okombo, who's on a secondment at MMV. He comes to MMV from having worked at Columbia and as a collaborator in David Fiddick's lab. We're also welcoming Caroline Lowe, who's a distinguished researcher at Isomorphic Labs in the UK, and Elizabeth Winzeler, who comes to us from UCSD, where she wears many hats, including professor in the Division of Host Microbe Systems in the Department of Pediatrics. She's also the Associate Dean for Research and Innovation in the School of Pharmacy and Pharmaceutical Sciences, as well as the co-director of the Malaria Drug Accelerator Program. So now Jeremy I want to start by posing the question to you. He talked about the 3 different ways you could feel about AI, but he didn't actually ask you how you felt. Well, I would suggest that there are 4 ways to think about it. You could be excited, uncertain, definite skeptic, or depends on the tool and the day of the week. So raise your hands. Who's excited? I knew you would be. Okay, great. That's good for excitement. A little uncertain? Okay, good. We're honesty out there. And definite skeptic? Okay, good for you, Diane. And then I think, it depends where I am, excited or uncertain, depends on the tool. I'm going to be it depends on the interface and the question at hand. There are things that ChatGPT tells me that I don't question at all, and other things it spits back, and I'm like, oh, I'm not sure. Okay, but I think mostly we're excited. I want to talk with our panel about how they have seen these tools help evolve how they do science and how they're approaching the work that they do. So John, can you start with us by describing how you're using artificial intelligence and machine learning in the work you do now, and contrast that to earlier in your career?
Thank you, Mary, and thank you for the organizers. Thanks, team, for the invitation. Just as a journey, starting back when I started malaria research, 2008. Mm-hmm. So I think we relied heavily on curated genetic and chemical structure databases. So if you think about it, the Plasmodium falciparum genome had just been sequenced in 2002. PlasmoDB had just been created around 2001. And then when you come to 2008, the Vivax genome had just been sequenced, and then ChemDB 2005. So this is what we relied on at the time, and it was data-driven, but hardly machine learning or hardly AI. And then in the last 15, 20 years, there's been explosion of data. So if you think about the thousands and thousands of whole genome sequencing on clinical isolates, and if you think about high throughput screening of drugs. And so in my work, having to appreciate all this and both handle and interpret this kind of complexity has been exciting, I must say, while at the same time I fall in initially as a skeptic, mm-hmm. with someone who's, integrating all this in the work that I do. And so as time moved forward and trying to answer the scientific questions that we ask every day, I am born and bred part medicinal chemist, part biologist working on antimalarial resistance. We have had to integrate this new reality in handling data at scale in the work that I do. So I don't just look at single mutations or single pathways, but rather holistically and integratively, if that's a word. So going forward, it's an excitement, I must say, just getting into the space that Jeremy was trying to describe. But with what David said, I don't think it will replace the traditional insight of what science is, but rather amplify it and give it an acceleration that it really needs. So I'm excited about the predictive power that's now in our hands as we try to figure out how best to interpret all this and make sense of the data and the tools that we have in our hands.
Okay, thanks. So Elizabeth, can you comment from your perspective?
Oh, okay. So one of the things that we've been doing in the Malaria Drug Accelerator, and this has been going on for many years, is we use genetics and evolution in many cases to find new targets. So everyone in the room probably knows that we'll have a molecule that's active in a phenotypic assay, and we want to know how it works. And what we typically do is we raise parasites that are resistant to this molecule, and then we sequence a genome, and then we look at these genome sequences and we find mutations, and this often will lead us directly into the target. And we've had many high-profile publications that people have probably read, where we discovered PFATP4 or PF-CARL or things like that. And we use statistical approaches to do it, but it doesn't always really work out as nicely as the publications show. And there's a lot of cases out there where we did this and we got a bunch of sequence and we couldn't figure out what was going on, and we just kind of put that on the back burner and then walked away, and sort of, this happens more than you think about. And the question is, so what happens typically is we have too many candidates and we don't have divergence on a clear target, and we have something different happening in all of our different selections. And what we want to do is to start using machine learning on the successes that we've had so that we can pick out that allele that is probably driving resistance. And so we've been compiling all of the data about new alleles that have come up through the Malaria Drug Accelerator and putting it together so that we can create machine learning models to try to identify which mutations in the genome are probably driving resistance when we get too many to look at. And so we've started to see things like they're in conserved areas of an alpha fold structure. They're more likely to transition to a bulky amino acid. They're next to a pocket. They're particular types of A to T transitions. And so we can put all of this together to create machine learning models that we can then run through genome sequences to say, oh, that's probably the mutation that's driving resistance. And this is potentially going to have applications as we move to looking at field sequences as well. So that's one of the cases where we're using machine learning in our own work to help accelerate drug discovery.
Great. So Carolyn, she mentioned AlphaFold as she's thinking about the relationship between resistance and structure. So from your role in Isomorphic, can you comment on AlphaFold?
Yes, thank you. So I had the joy of joining DeepMind 2 weeks before the CASP announcement, the big reveal where they declared that protein folding had been solved. Well, more or less. So I got to look at these structures. I was probably the first drug discovery scientist to look at these structures, and you could see where the ligands bound or where ions bound and where the zinc fingers were, and it was absolutely fascinating. And we've taken that technology into ISO and built upon that. And one of my favorite things to do is, for new starters, is to say, right, take something which you really system that you really understand. Now, all you have to do is feed in the protein sequence and the SMILES string, which is just a text representation of the molecule, and see what you get out at the end. And the number of times you'd get just gasps is a really nice piece of feedback. So we're trying to make platforms that everybody can use. We have released AlphaFold 2 and AlphaFold 3 into the wild. AlphaFold 2 structures are available on a database held by the EBI that everybody can use. AlphaFold 3 is available for all academic users, and that tells you where the ligand binds, where the ions bind, what happens when you do a post-translational modification. So there's lots of things that you never used to be able to do before. You had to wait for somebody doing a 3-year PhD to crystallize the structure for you before you knew what was going on, and now you can get an answer in 10 seconds.
Now, you had, in a previous discussion, you'd you were thinking about, can you look into use statistics or what the deployment is or the uptake within the African continent and academics there?
Yes, I asked the folks at Google to dig out some statistics. They're monitoring the IP addresses of people using these things. For low middle income countries, there's probably about top 27-30% of the users come from LMICs. In Africa, it's much lower use, so it's between 2 and 3% for both the database and AlphaFold 3. These things are out there. You only need a web browser. You don't need any complicated computer infrastructure to use these, and AlphaFold's not the only technology out there you can use. You can use all these things for free, and I strongly encourage you to have a look and see what you think.
Great. So, Jeremy, we've talked about biology and protein structure, and you've touched on the tools that you're developing around drug-like predictions and predictions of properties as people are designing compounds. Do you want to share any more thoughts? I expect you have some more thoughts on how MMV's thought about this enablement over the course of years.
So I am a believer in model-informed active learning, and this is a lot thanks to the experiences with these guys. We've taken data very seriously, and Dom, who's in the room, who is our data manager, right from the get-go has been focused on data quality, curating data in the right way. And that's put us in a fabulous position to then be able to actually model, those data. And actually, the ability to use models with virtual structures that so you plug your virtual structures into these models to get predictions. This is really, really powerful. And even, I guess what I've learned is that even if a model is poor, through active learning, the model can get better and be of use for the project. And even, and nudge you gradually and, forcefully into the right area of chemical space ultimately necessary for a candidate drug.
Great. So this takes us to a transition, the underlying data. Dave alluded to this in his talk, and I know, Elizabeth, in your role at MALDA, you are responsible for collation, curation, integrating lots of data. So what are your thoughts in terms of volumes of data and how to manage it and create the creation of the quality data sets as Jeremy's described?
Well, I'm not sure I was responsible for it, but I've always recognized the importance of it. So I've always gone out of my way to try to collate and compile the data, even though it wasn't necessarily apparent what the data would be used for in the future. And so one of the things that we've been doing, sort of it started as a side project, is to try to collect all the genome sequencing data that have been collected at MALDA, but also collected at MMV, and that have come from David Fiddock's lab, so that we can put all of it together and try to see something that that it's bigger than what we could get from one particular data set. And we've also mined the literature for a lot of these data sets as well. So I've always insisted, actually, maybe you might know me as a reviewer who asked to see the genome sequence deposited in the short read sequence archive. And And the reason I want them to do that is so that I can grab that data later on. And so then we can put it together and start creating data sets where we have every single mutation displayed on an AlphaFold structure that's ever been observed in the literature. And this is going to be the data set that will allow us to then build resistance risk into the compound design. And but the problem is it's really hard to actually get all of this data. And you know, the one thing that I've heard is that 90% of bioinformatics is janitorial work. And you know, just pulling it together, collating it, and the funders need to know that this is important if you're going to use artificial intelligence to, you know, to facilitate this type of data collection so that we can use it in the future.
I think of facilitation of data collection both in funding the acquisition of the data, right? How many IC50s are you funded to generate? And then how that's curated, who oversees the input and curation of that data? Who on staff, or what are the mechanisms and processes that you can put in place? Is that your experience too?
Yes, I mean, it's really challenging to get everyone to curate their data. I mean, I deal with students all the time. They don't really want to put it into CDD Vault. It's painful to learn how to do that, and but it's so important in the end to be able to pull it all out and to, you know, to see the big picture.
Yeah.
Yep. I mean, big pharma has a big advantage in that they can insist on people doing this, and I think a lot of the a little harder with students sometimes, but yeah. The only problem is that their data sets aren't usually publicly available, and that's where academics, you know, can be useful.
Yeah. Sounds like we have some heated agreement happening over in the corner here, but Dave, you noted in your presentation about you hadn't known much about malaria about 4 years ago, but Jeremy and the data from MMV really facilitated the engagement from Exscientia and now Recursion. So can you talk to us about what you saw in that and what the benefits, you know, what were the positive attributes that you recognized?
Yeah, absolutely. I think it comes back to this point about data quality. So for those people who are as old as I am in the audience, let's just walk back to a time before AI/ML. There's a publication, I think, either from BI or BMS that basically that highlighted that without malintent, about 80% of the world's literature, the really important information in those primary publications, could not be reproduced in another person's laboratory. So that's nothing to do with the evil of AI and ML or the skepticism. It's kind of, so it tells you that the data sets that ultimately we're all relying on, i.e. the public data sets, trust them but verify them every time. It tells you about how difficult it is to reproduce those materials. So if you're trying to build any kind of model, whether it's a model in your head off the data or an AI ML model, ultimately, yeah, you're looking at for patterns and reproducibility in that data. And that's the criticality behind this. And so I think the reason that kind of the MMV was so important is that when we're coming into this, it's like we can get access to most of that data because it's freely available, but we had no idea how much of that was reliable and and how much was reproducible. And so kind of the MMV team allowed us to kind of curate that data, understand the data from which we should trust, which data we should be more skeptical about. And then that underpinned the kind of the models we built. And therefore the models that, even though I agree with Jeremy, and kind of one of Jeremy's advisors, a former founder of Ngcete, basically, and he's right, a poorly performant model is better than no model at all when you've got active learning. But yeah, this all comes back to, it doesn't matter, I joke sometimes, and I'll share that joke here. Imagine, we all use ChatGPT, don't we, I guess? So imagine a world where ChatGPT is built on, imagine this, on conspiracy theory website data. That's kind of Truth Social, really, but anyway, we'll put that to one side. But if you then query that data, imagine what you get. It's exactly the same. It doesn't matter how, whether it's ChatGPT 3, 4, 5, or version 19. Those large language models are brilliant, but they're only as good as the data that sits underneath them. So I think it just re-emphasized the fact, like, every piece of data. DepMap, even DepMap for people who work in cancer, it's a pretty good data source, but it's not entirely always reproducible. So this is about really producing very robust, tightly reproducible, across labs data. If you can trust that data, you can build models from it, and you can do good science from it too.
So Jeremy, can you comment on when you try to build models and you need quality data, when MMVs tried to push the boundaries of what is available in the tool, say for PK predictions, how did MMV go about making sure the models they were building were robust? What was the underpinnings in the creation of MMV Sola and MMV Free?
So MMV Solar and MMV Free are really based around the state-of-the-art knowledge of pharmacokinetic equations. And so I would say that's a rather controversial step that we took in terms of those particular approaches. In terms of just models generally, if the models are poorly, if they perform poorly, or if indeed you don't have much data, all that we've learned is that we need a certain number of compounds and data before we can actually build a model that really has any use. But with active learning, this is something where the models can improve over time.
Thanks. We're going to close with a lightning round of question that I'm going to pose to you. So can you give an example of how your organization is leveraging partnerships for drug discovery, particularly with researchers in global health? And so, Carolyn, I'll start with you.
I think I've kind of already given you mine, because it's basically that you can access all the AlphaFold 2 structures and all the AlphaFold 3 structures, and please do go out and use them.
Great. Elizabeth.
Well, you have to form partnerships because there's limited resources and you really can't do everything yourself. And therefore, partnerships are absolutely critical in the malaria space. And so we have to rely on everyone. I can't do it all. So we build up expertise. David's the MIR expert, for example. Susan Wiley is the expert in proteomics. And we each have our own secret superpower that allows MALDA to work more effectively.
So, John, you've had an opportunity now to have kind of had a vantage point from several places, MMV, MALDA, FIDIX Lab. What's your take on it?
Yes, so I think as every speaker before me here has articulated, drug discovery, whether malaria or whatever aspect of global health is collaborative. I don't think any organization or institution has the infrastructure to do it by itself. So as you point out, it's a vantage point because I come from David's lab, which itself at Columbia, we're part of MALDA, and that itself is a huge consortium where there's a lot of collaboration with pharma, academic institutions. So within MALDA, the contribution that we put in there, just by example, there's a piece of work that we recently did with Elizabeth's group and the whole MALDA predictive features of the druggable plasmodium genome. And then just coming into MMV and being part of the MMV community and contributing to that as well, bringing in the drug resistance expertise as well as the biology expertise. But also I want to give a big shout out to the work that we're doing or that we've done previously with UCT and H3D, which itself, Kelly's here and the group is here. They have a very vibrant computer-aided drug discovery team, and which we collaborate quite closely. A lot of work that we've done with them, we're proud of that. So in a nutshell, these partnerships or these collaborations have kind of just, put us in a place where we look at drug discovery in a collaborative way, and that is just the only way it can be accelerated. So, yeah.
Yeah, thanks. And Dave, how about you?
I think if you live by this idea that basically that ultimately what we're all trying to do in this room is make drugs abundant and available to everybody. That doesn't mean everybody that's got money. Doesn't mean everybody that's kind of living in Europe or United States. It means around the world. And so you can't just kind of concentrate on like oncology or inflammation. You have to go after all areas of unmet need, tuberculosis, kind of malaria, other diseases. And so it's always been really important to our organisation that, yeah, we're commercial, and we make, and we're public, we don't deny that. But it's always been important to us that we try and kind of use our capabilities and support, diseases like malaria. And so we're proud that the Gates Foundation are shareholders in Recursion. We've given back model systems back into the environment outside of those. So partnerships are important, is that, as I said at the start, Recursion is not an expert in everything. We've got an infrastructure, but we're not malaria experts. We're kind of not experts in some rare diseases, but we have a capability where we can help. And I think it's on all of us. If we truly believe that when we talk about putting the patient first, then yeah, let's walk the walk. Yeah.
So I've come to Jeremy last because in the time that I've known Jeremy, he is the person who I would characterize as the one who wants everyone to come to the party. How can he create tools to have everyone have an opportunity to do this? So now that I put you in that light, Jeremy, tell us what shaped your thinking as you built those tools and the importance of collaboration and partnerships to you.
Well, we're MMV. Everything we do is in collaboration. We seek the best possible projects and partners, and we want to, work with those. We want to collaborate to deliver. And if that involves the need to develop our own thinking and to equip our teams, that's what we do.
Great. Well, thanks. Can you join me in thanking our panel? And so for our last session of the day, we don't get a coffee break beforehand, but I will invite Marcel Tanner to come up and offer his remarks.